Static pressure, we said that this favors filtration, about 60 millimeters of mercury, and this is due to the high resistance of the efferent arteriole. So again, going back to our two vessels here, our afferent arteriole and efferent arteriole. Now, because of the resistance that we see with the efferent arteriole, meaning that the radius or the circumference or the lumen of that efferent arteriole is smaller, that's why we see more of a higher glomerular capillary hydrostatic pressure, because as we can see here, the radius of the efferent arteriole is smaller when compared to the radius of the afferent arteriole. So we have a higher hydrostatic force present within the glomerular capillaries. The Bowman's capsule oncotic pressure, we describe that as being zero. There's zero protein in the filtrate. The forces that oppose filtration, the Bowman's capsule hydrostatic pressure. So this is that push force from the Bowman's capsule back into the glomerular capillaries. This is about 50 millimeters of mercury. And if we compare this to systemic capillaries, it's a bit higher. So if we think back to the interstitial fluid hydrostatic pressure, which is the force that opposes filtration in our systemic capillaries, it's much lower than 50 millimeters of mercury, right? And that is because there is a large volume of filtrate in a much smaller space here. And then when we look at the glomerular oncotic pressure, it's about 29 millimeters of mercury. Again, it's higher than what we see in the systemic capillaries because we have more protein present in a smaller amount of blood here. So think about it. If we're filtering all of this plasma across to the Bowman's capsule, all of the protein that's not being filtered with the plasma means that there's more protein sitting around here in the glomerulus. So as a result, we see a much higher glomerular oncotic pressure than when we look at the systemic capillaries. When we look at here, we see more protein present, so a higher oncotic pressure in the glomerular capillaries. Okay. All right, so here is an illustration of the calculation we just did. Again, make sure you are able to repeat something like this, even with different values. Make sure you understand why the forces are favoring or opposing filtration. Let's say I tell you that there was a tear in the filtration membrane and protein began to spill across into the Bowman's capsule. You should be able to tell me which pressure would be influenced, okay? And that would, of course, be your Bowman's capsule oncotic pressure, which is pulling fluid to the Bowman space. So if you've got proteins here, that would no longer be zero. So you should have that kind of understanding of these pressures and of what's creating these pressures in the different spaces. Okay, let's talk about some terminology. So we're going to describe a few terms, and we're going to talk about the values and how we calculate these different terms. So the first one is the glomerular filtration rate. We kind of introduced this already. This is basically how much plasma is filtered across the filtration membrane per minute. And we describe that value as being 125 milliliters per minute, or that worked out to be 180 liters per day. So that is the quantity of plasma that is actively filtered across our filtration barrier. We can also speak about another value, a renal plasma flow. So the renal plasma flow is how much blood is actually being circulated into the kidneys. So not how much is being filtered, but how much is coming through the kidneys to begin with. All right, and that is a value of about 625 milliliters per minute. So as we described here, not all of the blood that comes into this afferent arteriole is being filtered across. So 625 milliliters per minute of plasma is being moved into the kidneys. Of that, only 125 milliliters per minute is actually being filtered, right? 500 milliliters of that is going to exit via the efferent arteriole. When we compare those values to systemic capillaries, we know that it's much less, right? We should remember at this point the net filtration of about 3 liters per day. So every day, about 3 liters of fluid moves from our capillaries, all of our systemic capillaries, across into the interstitial fluid compartment. And in the kidneys, we see that much more filtration is taking place than what happens in our systemic capillaries. So if we compare this 3 liters per day to this 180 liters per day, we can really get a sense of that drastic difference. This brings us to speaking about filtration fraction. So the filtration fraction is looking at the GFR as a part of the total renal plasma flow. So we said GFR is about 125 milliliters per minute. Total plasma flow, meaning all the blood, all the plasma that is entering the kidneys, is about 625 milliliters per minute. And if we divide that GFR by the renal plasma flow, that is about 0.2 or 20%. So that is our filtration fraction. And what this value really means is that only 20% of the plasma that enters the kidneys is actually filtered across to into the Bowman's capsule. Okay, so 125 divided by that 625 is about 20%, and that is our filtration fraction, the volume of plasma that is actually being filtered. Another concept we want to talk about is the filtered load. So the filtered load is how much of a particular substance or particle or molecule is being filtered. And that is a product of the GFR multiplied by the plasma concentration of whatever that solute, whatever that particle is. So GFR multiplied by the plasma concentration of molecule X is going to give us the filtered load of molecule X, okay? So the quantity of that particle that is being filtered is the filtered load of that particle. And if we take glucose as an example, glucose is freely filtered across the filtration membrane. So we multiply the GFR, 125 milliliters per minute, by the plasma concentration of glucose. Now, in a normal healthy adult, there's about 100 milligrams per deciliter of glucose in the plasma, and we can convert that to 1 milligram per milliliter. And if we multiply that 1 milligram per milliliter, which is the plasma concentration, by the GFR, which is 125 milliliters per minute, that'll give us a filtered load of glucose of 125 milligrams per minute, okay? And I hope that that was clear. So basically, if you want to figure out how much of anything, how much of any molecule, how much glucose, how much protein, well, not protein, because protein isn't filtered, amino acids, anything else, how much of that molecule is being filtered, we simply multiply our GFR by the plasma concentration of the molecule in question. Okay, let's speak now about the regulation of GFR. So GFR is very heavily regulated, meaning that we do not want our GFR to be kind of moving around. We don't want it to be going up or down. We really ideally want GFR to stay at that 125 milliliters per minute. So we filter about 180 liters of fluid per day. And of that filtered volume, only 1.5 liters of urine is excreted per day. So in a healthy adult, less than 1% of the total volume that is filtered is actually excreted as urine. And what that tells us is that most of that fluid, 99% or more of that filtered fluid, is actually reabsorbed. That's going to be an important part of maintaining our fluid volume, okay? So if we were to completely eliminate all of the volume that was filtered, we would be grossly dehydrated. We would lose a lot of volume every day. And so to prevent that, the kidneys reabsorb most, 99% of the volume that is, of the volume of fluid that is filtered, and only eliminates less than 1% of that total volume. And that really leads us to understand that GFR is heavily regulated, and we want to keep the GFR at about 125 milliliters per minute. So this graph is showing us the GFR on the x-axis, the mean arterial pressure. So we can think about this as being our blood pressure on the, on the x-axis, and the GFR on the y. So we can basically compare these two values and see that if mean arterial pressure is changing, if it's changing in between 80 millimeters of mercury to 180 millimeters of mercury, the GFR stays the same. The GFR stays at 125. And that is because GFR is regulated. And when we say regulated, the body wants to maintain GFR at a set amount or a set quantity, even in the face of changing blood pressure, even in the face of changing mean arterial pressure. So a good example here is, think about you being dehydrated. Let's say you're grossly dehydrated, you're sweating profusely, you're hemorrhaging, and your mean arterial pressure drops low, right? Let's say it drops, but not below 80. Then the body is going to bring about certain mechanisms to raise the GFR so that it stays at 125. On the other hand, let's say you have mean arterial pressure that's rising, right, up to 180. The body would otherwise try to manipulate GFR. There are certain mechanisms that come into play that help to bring that GFR back down to 125 milliliters per minute. So there are three mechanisms that I'm going to talk about on the next slide, and these are the mechanisms that the body uses, that the kidneys use, to keep GFR at 125 milliliters per minute, even in the face of a changing mean arterial pressure within the value of 80 to 180 millimeters per mercury. Now, let's say we go lower than 80 millimeters per mercury or higher than 180 millimeters of mercury, then under those circumstances, our GFR will begin to change. So if we go lower, let's say we're completely hemorrhaging, our mean arterial pressure has dropped below 80, then the kidneys are going to take a hit and the GFR will fall. And on the other hand, if our mean arterial pressure rises above 180, think about really high chronic hypertension, chronic high blood pressure, the kidneys again are going to take a hit and GFR is going to start to rise. So let's talk about those mechanisms. And I really want you to compare, not compare, but to connect these three intrinsic mechanisms to this graph. These are the mechanisms that keep this GFR at 125, even in the face of changing mean arterial pressure. So the three mechanisms are myogenic regulation, tubular glomerular feedback, and mesangial cell contraction. And these are called intrinsic regulation, or intrinsic methods of regulating GFR. So let's sort of describe these here one by one. So myogenic regulation is basically a very similar phenomenon to what takes place in arterioles. So it's the smooth muscle that's in the wall of the afferent arteriole, and this is sensitive to increases in blood pressure. And upon having an increase in blood pressure, it's going to stretch. The cells of this afferent arteriole are going to stretch. And when they stretch, the response is to decrease the amount of flow that's coming in here, okay? So basically, if we have high blood pressure coming in, lots of plasma flowing into the afferent arteriole, that's going to cause a stretch on the afferent arteriole wall, the smooth muscle of this wall, and that is going to result in a contraction of those cells, which regulates or bring the blood pressure back down, okay? Or brings the amount of flow that's coming in back down. And what that really means to do is that means to keep GFR constant. Again, if I have a high mean arterial pressure, if my blood pressure is high, I don't want my GFR to change. So the afferent arterial cells are going to respond by constricting down on themselves in an effort to keep my GFR constant, even if my mean arterial pressure rises. The second mechanism, called tubular-glomerular feedback, is brought about by the macula densa cells. So the macula densa are a group of cells that are lining the distal tubule here. And let me go back here just so that this anatomical relationship is clear. So what we see here, this is the nephron, right? This is the Bowman's capsule. We can see that coming in between these two arterioles, so we've got the afferent arteriole and the efferent arteriole, and lying in between these two arterioles is the distal part of the tubule, right? So this relationship is really important to understanding the mesangial cells and how they bring about tubular-glomerular feedback. So the distal part of the tubule kind of wedges itself in between the afferent arteriole, which we can kind of differentiate here because of its larger diameter, and the efferent arteriole, again, which you can differentiate because of its smaller diameter. So that relationship is what brings about tubular-glomerular feedback. So here we can see that part of the tubule, so the distal part of the tubule. Some textbooks will tell you that it's the nephron. Some textbooks will tell you that it's the distal tubule. For our purposes, we're going to understand it to be the distal part of the tubule, okay? And so the mesangial, the macula densa cells that are found here are sensitive to the amount of flow that's coming into the distal part of the tubule. And what they do is they release or secrete certain paracrine factors that are going to travel to the nearby cells of the afferent arteriole and influence the afferent arteriole to either constrict or to relax, depending on the amount of flow that's coming through the distal part of the tubule. So a good scenario here is that I've got lots of flow coming through my glomerulus. That translates to lots of tubular fluid coming through the distal part of the tubule. And the macula densa cells are going to detect that there's increased tubular fluid or increased flow in the distal part of the tubule. And they're going to release certain paracrine factors that are going to make their way across to the afferent arteriole cells, the smooth muscle cells here. And that is going to influence the smooth muscle cells to contract or squeeze down or increase the resistance here. And that will decrease the amount of flow that's coming into the glomerulus, which will result in a decrease in flow of the fluid that eventually comes to the distal tubule. So the tubular-glomerular feedback is how the tubular structures can sense the flow rate, release factors that make their way communicate to the smooth muscle cells of the glomerulus, of the afferent arteriole here, influence the constriction of these smooth muscle cells, which will in turn decrease the flow that's coming through the glomerulus, decreasing the flow that eventually makes its way to the distal part of the tubule, and then that will turn off that signal or have a negative feedback effect on the macula densa cells. Alrighty. The third mechanism, or the third mechanism of intrinsic regulation, is called mesangial cell contraction. So mesangial cells are cells that are kind of studying the capillaries that are inside the glomerulus themselves. So the mesangial cells, I like to think of them as having a similar effect as the pericapillary, the precapillary sphincters. So one of the modifications or one of the mechanisms that you would have talked about with arterioles was the precapillary sphincter. So these strips of smooth muscle that can either constrict or relax and change the rate of flow that's coming through. And the mesangial cells have a similar role. So they're going to be lining around the capillaries of the glomerulus. And if we have lots of flow coming into the glomerulus, they're going to constrict to help decrease that flow and regulate the GFR. If we have a little bit of flow coming through the glomerulus, they're going to relax and allow more fluid, again, increasing the GFR and regulating it as well. Okay, so if there's a drop in GFR, these cells are going to result in an increase in GFR. If there's an increase in GFR, these cells are going to result in a drop in GFR. Again, all in an attempt to keep GFR constant at that 125 milliliters per minute, even in the face of increased mean arterial pressure or increased flow coming in here because of high blood pressure or low blood pressure. All right. So it's really, this is another important point today. So 2 important points that we talked about today that I really want you guys to take home. Understanding the Starling forces, what favors filtration, what doesn't, and understanding these 3 mechanisms. This is going to be a really important part of understanding filtration. Now I want to talk a little bit about this juxtaglomerular apparatus or juxtaglomerular complex. Now I don't want you to be too caught up with this, but I do want you to understand what it comprises of. So the juxtaglomerular complex is made of the macular denser cells that we just talked about. So the cells that are lining this distal part of the tubule. The extraglomerular mesangial cells. So the extraglomerular mesangial cells behave just like the mesangial cells of the glomerulus. The only difference is that they are outside of the glomerulus, right? Hence the term extraglomerular. And then finally, the granular cells of the afferent arteriole. And I just want you to have an understanding of these 3 components of the juxtaglomerular complex, because we're going to come back to this not in a whole lot of detail, but I do want you to know that it exists when we talk about renin later on, because renin is secreted by the cells of this complex, okay? All right. Now, these 2 flow charts are 2 examples of applying the intrinsic mechanisms, or 2 of the intrinsic mechanisms that we talked about. So on the left, this is the myogenic regulation, and we'll kind of go through this here. So let's say I have an increase in mean arterial pressure, right? Let's say I have hypertension, or I'm under stress for some reason, and my mean arterial pressure goes up. The kidneys would be tempted to decrease GFR. And so what we don't want is GFR to drop just because my mean arterial pressure went up. And so the myogenic regulation is attempting to bring GFR up and prevent it from dropping in the face of an increased mean arterial pressure. So let's walk through how this happens. So pressure rises, there's an increase in the pressure that's coming in the afferent arteriole. That is going to cause a stretch on the smooth muscles that are lining the afferent arteriole. The result is going to be, or the response of those smooth muscles is going to be to constrict down on themselves or increase in their constriction. Now, when they increase in their constriction, that is going to increase the resistance that is coming through that afferent arteriole. And when we say increase in the resistance, increasing the impeding flow, right? Remember, resistance is the impediment to flow. So as we clamp down on the circumference or the diameter of that arteriole, we're going to increase the resistance in that vessel. That would otherwise result in a decrease in glomerular capillary pressure, right? Remember, as we clamp down on that vessel, the tendency would be for glomerular pressure to decrease. But that doesn't happen. What happens is glomerular capillary pressure is going to increase. Okay, as those smooth muscle cells begin to increase their resistance, the glomerular capillary pressure is going to increase. That is going to increase the glomerular filtration pressure, or the amount of pressure that's coming into the afferent arteriole, which will increase GFR, and that would prevent the otherwise expected drop in GFR. Okay, I hope that makes sense. It's a little bit counterintuitive to kind of understand. So I'll go through this one more time. So if I have an increase in my mean arterial pressure, the kidneys should result in decreasing my GFR. But I don't want that response. So the myogenic regulation, right, the smooth muscle that is lining those afferent arterioles is going to constrict, which is going to increase the resistance, which is going to increase the pressure that's coming in. And that is going to increase my GFR and prevent or negate a drop in GFR. That's going to bring GFR back up to 125, which is keeping it constant or stable. Okay? So in this scenario, the stimulus would be a rise in mean arterial pressure. The natural response would be a drop in glomerular capillary pressure, which would cause a drop in GFR. But the myogenic regulation is going to correct that and cause a rise in glomerular pressure, which would cause a rise in GFR, and that will keep GFR constant or stable. Alrighty? Okay, let's look at the tubular glomerular feedback. So very similarly, the initial stimulus would be a rise in mean arterial pressure. That's going to cause more pressure coming into the glomerular capillaries. That's going to result in an increase in the amount of pressure coming into the glomerular capillaries. And what that would cause is a drop in GFR. If we've got more pressure coming into the capillaries, the capillaries are going to increase their constriction, they're going to increase their resistance, and that would cause a drop in GFR. But because we have this tubular glomerular feedback system happening, that is going to release paracrine secretions or paracrine factors that are going to communicate to the afferent arterial cells, and that is going to allow them to constrict on themselves and increase the pressure, which will increase the GFR. Okay, so again here, the stimulus would be mean arterial pressure rising. The natural response would be a drop in glomerular capillary pressure, which would result in a drop in GFR. But because of these paracrine factors that are detecting this increase in flow, that's not going to happen. That's going to instead cause these afferent arterial smooth muscle cells to constrict on themselves, which will increase the glomerular filtration, the glomerular capillary pressure, which will increase the glomerular filtration pressure, which will increase the GFR. Alrighty? Okay, so both of these scenarios are bringing about the exact same response. There's a rise in mean arterial pressure, which would otherwise trigger a drop in GFR, but we don't want that drop in GFR. So the response that is brought about is to rise GFR, and that helps to keep GFR stable.